Hello, good morning, student learners. Welcome to another class. Our topic for today is microorganism in action. Last week we talked about culturing, how microorganisms are being grown in special medium in the laboratory. And this culturing helps us to know the way they grow, their characteristics, their color, the sizes, different features like that in microorganism that has been cultured. Now today we'll be talking about how they act, their actions. Now microorganisms, like every living organism, do increase in size and multiply in number of cells using either culture medium, rather or suitable surface such as moist spread as source of food. So microorganisms, here we want to look at microorganisms, how they grow, their action. They grow like normal human beings. They increase in size. Their cells multiply like normal other living things. Rather, if environmental conditions of growth, such as food adequate temperature and humidity, are favorable, such as increase in mass and number of cells of the colony, is an index of growth of microorganisms. So, if you are in a favorable condition, there will be fast growth rate. Now, stages of growth in microorganisms, we have the lag phase, exponential phase, stationary phase, decline phase. Now, the lag phase is when a microorganism is introduced into the culture medium. It takes some time to adjust to its new environment, and it accounts for initial slow growth phase known as lag phase. So, when just like any other living things, when a living thing is placed in another environment, it will take time to adjust. Even human being placed in another environment take that human being to adjust. So that's an example of lag phase to microorganism. Now exponential phase. After lag phase, exponential phase proceeds with the maximum growth. At this point, there is increase. However, as population size increase, there is competition for food and space. Now there is competition for food and space, and when competition of food and space happens, this is us to the stationary phase. It will get to a point due to the competition for food and space, it will slow down. Because imagine two people eating a bag of rice for a month. Now, when 10 people eat a bag of rice for a month, you see there will be difference. Now, due to competition of food and space, the slow growth will fall down because it gets to say the food might not be enough for them. Some people will have to die and all that. It is only survival of the fittest that will be able to make it further. Now, until the population cease to grow, thus leading to stationary rate. So to get to a stage to be stationary rate, it will stay, nothing is growing. So, and they are competing due to the competition for food, for the food available, it's not enough for them anymore. Now decline phase, as toxic waste increases and the food decreases, the food will keep decreasing as population increases now, and toxic waste increases, the cells stop dividing. Yeah, because those toxic waste are poisonous. Cells dividing, the dormant cell begins to die, causing a decline in growth rate. So all the cells begin to die, and as a result, microorganism dies, and there will be decrease in the growth rate of microorganisms. So these are the stages of microorganisms. See how they are being grown, so where they eventually die. Now, ways of measuring growth in microorganisms. We have two ways. You have the first method and second method. Now, a bacterial sample inoculated into a nutrient jar. As bacterial population increases, the clear liquid medium becomes cloudy or turbid. Hence, something turbid, something not clear. Progressive increase in turbidity increase a related increase in the number of bacterial cells. This property is used to measure bacterial growth. Now, that instrument used to measure turbidity, we know is tertiary disc, and here can be measured also with spectrophometer. Okay, now the second method is this is a method. In this method, small samples of bacteria are taken from the nutrient angle at regular intervals of time. Each sample is dilated several times. Each dilated sample is inoculated onto a nutrient agar medium in a petri dish and is counted. So you can go through that. Now, what are the benefits of microorganisms? Microorganisms also are beneficial and they are also harmful. The last time we talked about microorganisms, we talked about some that are pathogenic and some that are non-pathogenic. Those that are non-pathogenic are beneficial, while those that are pathogenic are harmful. Now, I want to talk about those that are non-pathogenic, their usefulness. The benefit of some microorganisms. Now, in nature, they helps in compost formation. Now, they are the ones that are responsible in the decay of dead remains, the saprophytes. Now, nitrogenous waste, they helps in nitrogenous fixation, rather maintenance of soil fertility. They increase the soil fertility, which helps for agriculture. Digestion of cellulose, they helps in digestion of cellulose in human. Decomposition, they helps you decompose any organic substances. Sewage making, sewage treatment, they also help in sewage treatment. So look at their benefit in nature. Now, in medicine, manufacturing of drugs and vaccines, some are used in manufacturing of drugs like yeast, sources of vitamin B, and sources of enzymes. Now, in industry, yeast are also used for baking cake, preparation of alcoholic drinks. Can also use yeast for preparation of alcoholic cake, making of cheese and yogurt, wetting of jute. It's not jute, it's jute. Curing tobacco, tanning of height and skin. Sorry, I will adjust it in the notes. It is height and skin. Harmful effects of some microorganisms. Cause of disease. Yes, they are the cause of disease. They are the cause of disease. Plasmodium and trypanosome. The cause of disease. Spoilage of food. They are necessary for spoilage of fungi. Dissemination of materials. They cause death in living organisms. Microorganisms like viruses, like COVID-19 and the likes, HIV, they are also microorganisms. Herbs, rickets, name them. They cause death to living organisms. Disease caused by microorganisms. The following are disease caused by microorganisms. You can go through it. Animal disease caused by virus, plant disease caused by viruses, animal disease caused by bacteria. Go through them. These are examples of the disease caused animal disease caused by virus, and these are examples of plant disease caused by viruses. Animal disease caused by bacteria, these are examples of animal disease caused by bacteria. These are plant disease caused by bacteria. Animal disease caused by fungi, these are examples. Plant disease caused by fungi, these are also examples. You can as well go through it. Animal disease caused by protozoa, trypanosomiasis, coccidiosis, trichomonias, and red water diseases. Malaria is caused by plasmodium. Don't forget, trypanosomiasis is caused by trypanosome. Coccidiosis is caused by Emereal species. Red water disease is caused by Babesia species. So you can read that. And spread of disease causing microorganisms. Microorganisms are spread through the following: can be through air, water, food, animals, or personal or direct skin contact, like the Ebola and the likes like that, and COVID. So, certain diseases known as contagious can be contracted by direct contact with infected person, e.g., leprosy, measles, tuberculosis, sexually transmitted diseases such as gonorrhea, syphilis, and AIDS. Food and waterborne disease. So these are the examples of food and waterborne disease. We have the disease, we have infected hepatitis, typhoid, malaria, poliomyelitis. We have the amoebic dysentery. We have the bacillus dysentery. We have the food poisoning. We have the parathyroid. So these are examples of food and waterborne disease, and their causes. Those organisms that causes them that have been listed. Their mode of transmission: is it by water, is it by food, by drinking water, and their host, where those organisms they really have those living things they affect. Those living things, these causative organisms affect are their host. Now, what are the symptoms you are going to see in their hosts for you to know that yes, truly these organisms have infected them? So these are the symptoms. Thank you. Do have a wonderful day. And if you have any question, we can meet in the classroom.